This lecture, we're going to look at the synapse itself. The synapse is going to be very important for drugs. And in this case, how do we stop the signal? How do we get the signal to keep going? And what we're going to look at enzymes that break down those neurotransmitters. So if we want the neurotransmitter to stop, we can produce more enzymes to get more neurotransmitters to leave the synapse. If we want the synapse, to have more neurotransmitters, then we'll just get rid of the enzyme that actually will stop the breakdown of neurotransmitters. So the synaptic knob is that last little bit. Presynaptic neuron is presynapse, so before the synapse, and then the postsynaptic neuron or target cell is going to be after the synapse. What it does, that action potential goes all the way down. This is a little tricky, but we're not really going to talk about it. Calcium channels open at the synapse. Once calcium enters the synaptic knob, it allows these vesicles that contain neurotransmitters to start to bind to the cell membrane. They get released into the synaptic cleft, this little space. And then there was something on that other slide called ligand channels. Ligand just means that if something gets released, you have a channel that it can bind to. So that's why it's a ligand regulated ion channel, because something has to bind to it. Then they open, they allow ions to travel through them. That's your synapse. A few more slides though. So we have about 100 different types of neurotransmitters. The main different types: acetylcholine. Acetylcholine is in its own class. It's formed by acetic acid and chlorine. Acetylcholine can either excite a muscle fiber or inhibit a muscle fiber, depending on what type of receptor it has. The amino acid neurotransmitters, you have glycine, glutamate, aspartate, and gamma aminobutyric acid, GABA. You've all either heard of GABA directly or indirectly. You have GABA blockers that can actually block different types of GABA channels. They're used for mental health and other types of things. So GABA, either directly or indirectly, you've all kind of heard of this amino acid type of neurotransmitter. Glycine and glutamate are very important in the eye, in the brain, and those are other two really important ones. You can have amino acids or monoamines, and these are amino acids that just remove their carboxyl group, and then they just have the amino group. You have epinephrine, norepinephrine, dopamine, histamine, serotonin. Those are all some you've probably heard of. And then other ones that are neuropeptides, which are just small chains of amino acids that can then be used for neurotransmitters. A neurotransmitter does not have the same effect everywhere in the body. That's going to be because a receptor is going to open or close depending on what neurotransmitter is going to touch it. So you can have multiple receptors for each particular neurotransmitters. Serotonin has 14 different ones. So you might have heard serotonin, dopamine. These are all good things for memory, for feeling good. But because dopamine and serotonin, they've been studied over and over again, serotonin has 14 different ones. So depending on what type of cell has what type of receptor, it's going to have a unique effect on that one cell. So receptor is going to govern the effect of the neurotransmitter on the target cell. Luckily for you, you're not memorizing any of that. So we're just going to look at the basics. Some are going to be able to excite the cell. Some are going to be able to inhibit the cell. Excitatory, like we mentioned before, makes the cell more positive towards threshold. Inhibitory makes it more negative or away from threshold. And the way it becomes more excitatory, Positive ions go in, make it more positive. Make it inhibitory, negative ions go in, makes it more negative. And then some effect depends on what kind of receptor the postsynaptic cell has. We'll look at that when we look at chapter 15. Some of them have open ligand regulated ion gates. Some act through second messenger systems. Not important for you, but if you have a ligand, the neurotransmitter binds to the receptor opens. If it's second messenger, the neurotransmitter binds to the receptor, then something on the inside of the cell will send a signal to the inside of the cell to maybe the DNA or some specific type of enzyme to work. So, 3 types of synapses with different modes of actions. The excitatory cholinergic, inhibitory GABAergic, and excitatory adrenergic. Using acetylcholine, using GABA, using, we'll talk about the different types of adrenergic receptors. But GABA, and these last 2 are probably the ones you will deal with mental health. So synaptic delay is the time of arrival of the signal and the axon terminal. And it's about a half of a microsecond for all these complex sequences to occur. So they're very, very, very quick. In the cholinergic synapse, you're going to have acetylcholine as a neurotransmitter. At the skeletal muscle, acetylcholine excites the cell. A different type of receptor is going to be found on the cardiac muscle. So if acetylcholine goes to the cardiac muscle, it's going to inhibit it or stop it. And this is how our body can either open and excite certain cells and inhibit others. So here you can see the basic one where it opens, goes down to the muscle cells, potassium flows out, excites it, sodium goes in, excites it, and eventually produces an action potential that goes down the T-tubule and allows calcium release into the muscle fiber. GABAergic. This one is using GABA as a neurotransmitter. The nerve signal triggers the release of GABA into the synaptic cleft. GABA receptors are chloride channels. Chloride is an anion, which means it's negative, enters the cell and makes the inside more negative than the resting membrane potential. If you make it more negative, you make it more inhibited, you make it further away from threshold. So this is the most common inhibitory neurotransmitter in the brain. So if there's chemical imbalances, there's issues with neuroscience or neurobiology, you're usually going to be using some type of drug that's either going to have an effect on GABA. If you have an overwhelming feeling of just sitting there, not doing anything, you're lethargic, you don't want to move. If you just block GABA a little bit, you won't have those inhibitory feelings anymore, even though they're very complex, a lot of things are happening. You block GABA, you then can have more excitatory reactions that will change whatever action or behavior you're trying to get at. And it's not that easy because it's complex. So adrenergic employs the neurotransmitter norepinephrine. Norepinephrine is also called noradrenaline. Norepinephrine and other monoamines, they're going to act through a second messenger system. Adrenergic receptors are very kind of confusing because what happens when norepinephrine binds, it's going to use a G protein. The G protein is then going to trigger cyclic AMP. Cyclic AMP is then going to work on an enzyme, and then that enzyme is going to create some action within the cell. So it's much slower than cholinergic and GABA, just because it has to spend some time for stuff inside the cell to happen. The issue with this, the advantage of enzyme amplification, which means if one signal comes into a cell, that enzyme can be amplified multiple hundreds hundreds of times within the cell and create a stronger effect than the other two receptors. So this is what happens. Norepinephrine goes on to it. This little thing called the G protein will activate cyclic AMP. Cyclic AMP has multiple effects. It can go into the nucleus and cause genetic transcription. If that's the case, it can cause enzyme synthesis. If it causes enzyme synthesis, it can cause metabolic changes. It can also just activate an enzyme which does the same thing, or it can kind of go back and open a channel for ions to go into it. So norepinephrine can do a lot more things depending on the type of channel that it's there. So postsynaptic potentials. Neurointegration is based on the postsynaptic potentials produced by neurotransmitters. So what happens to the cell with that neurotransmitter? Typical neurons have a resting membrane potential of -70, and their threshold is about -55. So if you have an excitatory postsynaptic potential, that means any voltage change in the direction of threshold that makes it more likely to fire. So excitatory postsynaptic potential, an EPSP. Some people will just use these abbreviations. So if you make it more closer to threshold, that's excitatory, that's exciting that postsynaptic neuron or cell. Glutamate and aspartate are excitatory in the brain and brain neurotransmitters, and they produce excitatory postsynaptic potentials. And glutamate is the major excitatory one in the brain. Inhibitory, on the other hand, make it less likely to fire. So any voltage that takes it away from threshold makes it less likely to fire. Neurotransmitter hyperpolarizes the postsynaptic cell, like GABA in the chlorine channel. It makes it more negative than resting membrane potential, making it less likely to fire. This is produced by neurotransmitters that open ligand-regulated chlorine gates. Inflow of chlorine makes it more negative. But wait, there's more. Glycine, GABA produce inhibitory postsynaptic potentials and are inhibitory. Acetylcholine and norepinephrine are considered excitatory in some cells and inhibitory in others. Just kind of know that, but we're not going to memorize which type of cells right now are inhibitory or excitatory. So here we go. Excitatory and inhibitory. If you make it more positive, it's excitatory. You make it negative, it's inhibitory. You see this, you're like, okay, that's pretty easy. Unfortunately, the cell just doesn't get one. It gets many of these. So one neuron is going to receive inputs from thousands of other neurons. Because I told you there are a trillion of these in the body, it's receiving a bunch. So the incoming nerve fibers may produce an excitatory, while others produce an inhibitory. Neuron's response depends on whether the net input is excitatory or the net input is inhibitory. That's called summation. And that's adding up all of these postsynaptic potentials and then seeing if it is going to fire because it goes to threshold or if it doesn't. So it's that delicate balance between the excitatory and the inhibitory that enables the nervous system to make decisions. So even though we're going to learn about parts of the brain and all this other stuff, it's all of this interaction between single neurons and different nuclei in the brain that allows inhibitories and excitatories, that yin and yang, to kind of see if it's going to go up and make an action potential or if it's going to keep it away from an action potential. And temporal summation occurs when single synapses generates excitatory postsynaptic potential so quickly that each is generated before the previous one fades. And I'll show you what that means. This allows the excitatory postsynaptic potentials to add up over time to threshold voltage that triggers an action potential. Sometimes it's going to be spatial. So several different synapses is going to add up to threshold. So let's take a look. So this is when one neuron is sending an intense signal on just one. And then that will create an action potential, and that'll signal it to go down the axon. This is spatial, where in space there's multiple neurons that are sending a signal. All of those will add up to threshold, and that will send the action potential down the axon. Temporal is one, spatial is multiple. And this is another look at it. So all you're doing at the trigger zone, or the axon hillock, those excitatory postsynaptic things can go as steps and just start getting closer and closer and closer to stimuli. And then that's your sodium channel opening, potassium channel opening, and then there's your normal action potential. And this is just another way to look at it, where you can have an inhibitory neuron right here that's not turned on, but right when that's turned on, that will produce an inhibitory postsynaptic potential and shut off that neuron. And these are multiple ways our body is going to be able to use either excitatory or inhibitory. Here's another one where it opposes it, but it's still the same thing. If it's off, you have activity. If it turns on, it'll shut off activity in that one synaptic knob. So let's look at a couple diseases before this one's done. Alzheimer's, about 100,000 deaths per year. 15% of the population is over 65, and then have it, and then 45% of the population over 85 have Alzheimer's. Memory loss, recent events, moody, combative, lose ability to talk, walk, and eat, pretty much live. This shows deficiencies of acetylcholine and nerve growth factor, NGF. What happens here is you have atrophy of the gyri, of the folds of the cerebral cortex, neurofibril tangles and plaques. You have formation of beta amyloid protein from breakdown product of plasma membranes, and one of the treatment is to stop the beta amyloid production. The beta amyloid has all these different side effects. So what happens is that you can give people cholinesterase inhibitors. So just look at that word. It's not on the test, but you will be asked a question later on about something like this. So you're going to be put on your doctor hat and kind of think. What is cholinesterase? Yes, enzyme, because it has ASE. It's an enzyme that probably breaks down choline or acetylcholine, right? So if you have a cholinesterase inhibitor, this inhibitor stops this enzyme from breaking down acetylcholine. If you have more acetylcholine, you have more excitatory response, so you'll be more likely to be less moody, less combative, ability to talk. And then these are those little plaques, those little buildup of tangles and everything like that. And then finally, Parkinson's. Parkinson's will look at one particular part of the brain called the substantia nigra, and we'll look at it. It's called nigra because it's darker color. When they saw it, they're like, that's kind of cool. And it was substantial, so that's why they called it substantial nigra in Latin. So in Parkinson's, progressive loss of motor function in the 50s and 60s, usually no recovery. They have gotten to the point where they can slow it down. And what happens is the dopamine releasing neurons in the brain, they start to stop releasing dopamine. Dopamine normally prevents excessive activity in motor centers, especially the basal nuclei. Involuntary muscle contractions, chewing, fidgeting, stuff like that. They have treatment and drugs and physical therapy. Dopamine precursor L-DOPA is able to cross the blood-brain barrier. Problem with that is it can damage the heart and the liver. You can have monoamine oxidase inhibitor, which dopamine is a monoamine, and it inhibits, an inhibitor slows the neural degeneration. And surgical techniques to relieve tremors, what they do is they go and they just make cuts and lesions to axons so you don't have as many lesions. There's many different ways that they can work on Parkinson's, but Parkinson's, dopamine, Alzheimer's is going to be those plaques.